Okay, we will talk about bioavailability and this recording and bioequivalence. Both of those terms, you know them. Excuse me. You know them already. We will talk about them in terms of kinetic. It's an easy subject. You know a little bit about it already, but let's see what you need to know more related to the calculation of bioavailability and bioequivalence. If we start with the definition of bioavailability and the 1977 by the US FDA, they defined the bioavailability as the rate and extent at which the active ingredient or the therapeutic moiety that we are interested in is absorbed from a drug and becomes available at the site of action. But this definition, so we are talking about rate and extent, we are talking about active ingredient of the drug or whatever that you are interested in and its availability at the site of action. This definition is not that correct or has some flaws because even we are interested in the rate and extent, even we are interested in the active ingredient, but we can determine how much or as extent or rate of the drug at the site of action. That's why the American Pharmaceutical Association definition is more accurate because it's talking about the amount of the administered dose and the rate at which the drug reach the general circulation or systemic circulation. So we are talking about the rate and extent of the drug reaching a systemic circulation, and we can quantify this. That's why this is more accurate as a definition. We wish that we could use this or quantify this, but actually the site of action or the receptor, theoretically we have uh, the pharmacology and stuff like that, but theoretically we have for each drug or for each moiety we are interested in that the drug is reaching the active ingredient in this percentage or that amount, but we can't quantify it easily. So that's why this is more accurate. We will talk about some definition related to the bioequivalence and bioavailability and how we can define those kind of things. And this diagram will help you in defining those definitions or terms. So let's play with it as yes or no questions. If you have two products with the same drug, if you don't have it, this is a question. If we don't have the same drugs, then we are not talking about equivalent products. So they are not equivalent. If we having the same drug, then we are ask ourselves if we are talking about the same amount of active ingredient in the same dosage form. If it's no, then we are talking about pharmaceutical alternative product, which means we are talking about the same drug, but we may have some differences either in the amount of the active ingredient or the dosage form. If we are having the same drug with the same active ingredient and the same dosage form, then we are talking about pharmaceutical equivalent products. Then we ask ourselves if we have significant differences in the rate and extent of absorption slash amount or rate reaching systemic circulation, not just absorbed, reaching systemic circulation. That's why, and I will just pinpoint this note when we are through the slides. It's not just the absorption, but also reaching the systemic circulation. If we have significant difference in the rate and extent, then we are talking about bioinequivalent product. But if we don't have that much of difference, then we are talking about bioequivalent product. By default, we are aiming that the bioequivalent products having are resulting in therapeutic equivalent, which means that within similar and not significant differences in the safety profile or the when the drug is effective and when the drug is toxic. So again, if we go back, if we are talking about same drug with the same amount of active ingredient in the same dosage form, we are talking about pharmaceutical equivalent product, then we do some studies to determine the rate and extent of the absorption of the drug or how much the rate and extent of the drug reaching systemic circulation. If we don't have that much of significance and the difference, then we are talking about bioequivalent product to get at the end therapeutic equivalent products. Okay, how we determine this, we will see that we will study different parameters that some of them, and we start to compare them. So to answer this question, if they are different or not, significantly different or not, we usually have the statistical analysis to know if they are significantly different or not, and there's guidelines for that. So if the area under the curve is different with this percentage, then we consider it significant. If not, we consider it not significant, and so on and so forth. So it's usually we do the studies, the kinetic studies for the product A and product B, and we see if they are significantly different using statistical analysis. Here we are talking about the definition of those terms that we just mentioned, but in words or word wise. For example, pharmaceutical or chemical equivalent products. So we are talking about this, right? Two or more drug products contains equal amount of the same therapeutic active ingredient in identical dosage forms and route of administration. So we are talking about, again, same drug, taking the same amount of the drug in the same dosage form. If we are talking about pharmaceutical alternative, then we are talking about this. Then we are talking about the same therapeutic ingredient, which is the same drug, but they are differing in the salt, ester form, or dosage form, or the strength. So we are talking about same drug, the same exact drug, but we are changing the amount or the excipient that we are talking about, or the dosage form. One example of this, when we are talking about controlled release drug versus conventional direct or rapid release formulation. When we are talking about bioequivalence, again, if you go back, bioequivalence, something related to this, we are talking about two or more equivalent product for sure. So we are talking about the same drug, same dosage form, same amount of the drug in the same dosage form that produce comparable bioavailability. The meaning of comparable bioavailability, not identical, but we don't have statistical significant difference between them as bioavailability. And when we talked about bioavailability, we said it's the rate and extent of the drug reaching systemic circulation, right? So for any drug when administered in equivalent dosing regimen. Bioequivalent drug products may contain different inactive ingredients or excipients for sure. We are mainly considering the same active with the same amount and the same dosage form, but it doesn't matter if we have different excipients. One of examples of the parameters that we usually compare when we are talking about bioequivalence or bioavailability, it's total area under the curve, CB max, and T max. And this is when we talked about this in the extravascular. We said that usually the area under the curve give us an idea about the extent of the drug reaching the systemic circulation. CB max, both the extent and rate, and T max, the rate of the drug reaching the systemic circulation. When we are talking about therapeutic equivalence, again, this is the last part of the diagram, which is this. When we are talking about the therapeutic equivalence, it means we are talking about pharmaceutical equivalent products, two or more, that produce the same efficacy and or toxicity in the same individual when they are administered in the same dosing regimen. So we are talking about the same or similar efficacy profiles or toxicity profiles. They are not significantly different from each other. For the bioavailability calculation, we usually have two types of bioavailability, the absolute bioavailability and relative bioavailability or comparative bioavailability. Let's start with the first one. Absolute bioavailability could be assessed or determined using either the total area under the curve or the cumulative amount excreted in the urine total, which is XU from 0 to infinity. We can use this or that. To determine this, if you remember, we need to have the concentration in the plasma or in the blood versus time. To determine this, we need the amount to be excreted in the urine. And we have the same notes about this in the urine, if you go back to the details when we talked about it, in the IV or the extravascular. We need a significant fraction of the drug to be excreted unchanged in the urine, and to have it characterized as much as we can to reach 0 to infinity. So that's why if we don't have significant fraction of the drug excreted, we said that this happens at infinity, which is usually, practically, we can't reach it. So at the beginning, you need, or firstly, you need the drug excreted unchanged in the urine, right? This is one thing. Another thing, we need a significant fraction of that to be excreted. To determine 0 to infinity, or the total amount excreted in the urine, this is practically cannot be done. So usually in practice, we have 5 half-lives, and this is what we mentioned earlier when we talked about amount excreted in the urine. To determine the absolute bioavailability using area under the curve, which in other words, we are using uh, plasma data, which is something very important. It's not a highlighter. So using the plasma data to determine the absolute bioavailability, we are using area under the curve. So we either use the plasma data or we use urine data. So starting with the plasma data, because it depends on concentration in the plasma, right, versus time, whatever that you are talking about, extravascular or intravascular. Okay, we are using this profile on rectilinear to determine the area under the curve. So to determine absolute bioavailability as extent or from the fraction of the drug absorbed slash reaching the systemic circulation, we determine it by having total area under the curve from 0 to infinity for the drug of interest over area under the curve from 0 to infinity, which is total again to the IV, or our reference, which is IV, because we know that this is the maximum that can we have in the blood. Okay, but we know from the equation that clearance, if you remember, for let's say IV equals dose over an initial dose over area under the curve total. So we know from this that area under the curve is proportionally, if we have it here, right? Area under the curve is proportionally related to the amount of the drug administered as a dose, which means that changing the dose would change area under the curve. So we need to normalize area under the curve to the dose of the drug, which means that we need to divide by the dose of the first drug, so area under the curve over the dose, over area under the curve over the dose for the IV. This is, let's say, either it's parenteral or sub-Q, oral, whatever, extravascular over IV. Again, we are using IV because it's the maximum that can we reach of the drug inside the body, because the drug is directly entering the plasma. Okay, so we need to correct, one of the main things that we need to correct each of the area under the curve to the dose, so we are talking about the same dose. If it's the same dose, you don't need to do this this step. But again, even if you do it, this will goes with this. Okay, so profile wise, if we are talking about IV bolus, CB versus time for IV, CB versus time for extravascular. So we are determining not only the concentrate, the area under the curve from 0 to certain time, which is your T last in this and T last in this. No, we are measuring it until the infinity, right? So we are having it from 0 to infinity, and the same thing for here. We are having it from 0 to infinity, which is profile-wise, it's like I'm comparing this to this, right? This is your standard and your equation here. So this is the profile of IV. So um, we are comparing this to total to the total of this again. Okay, this is profile wise, and we know that we can determine this using the trapezoidal rule from 0 to the last, then to t last or CB last. Then we can determine this from the integration, and the same thing here as we mentioned in the extravascular and intravascular chapters. For the urinary data, to use it from the urinary data, again, we use just the plasma data using area under the curve. We can use also urine data to determine the absolute bioavailability. The same thing, we are using total amount excreted cumulative in the urine of the drug that I'm interested in over the amount total from 0 to infinity to the IV, which is our a standard or reference, correcting that to the dose of each one of them, as I just mentioned. But again, if you have the data of this, which is usually not practical, you can use them. If I gave you this in the exam, just use it. But if we don't we don't have enough time, we need to have it from zero to certain time, and this time for each one of them, and this time should cover at least 5 half-lives of the drug, which means if you have the half-life of the drug, and I give you, let's say that the half-life of the drug is 5 hours, okay? If you have the half-lives as 5 hours for the drug, and I told you that we did the measurement for 10 hours, is it enough? No, it's not enough, because we didn't cover 5 half-lives of the drug, right? Sorry, let's go back to I don't recall what I just said. If we have the half-life as 5 hours, I'm not sure if I said this right, but if we have the half-life of the drug as 5 hours, then we did the measurement in the urine for 10 hours. Is it enough? No, it's not enough, and we can't use urine data to determine that, because we have only 10 over 5. We only have 2 half-lives covered, okay? I need at least 5 half-lives to be covered, so I need at least 25 hours to be covered in the measurement. For sure, what I'm talking about is just an example. So I need to know if the time that I'm covering here, 0 to certain time, the time I'm covering is covering 5 half-lives of the drug. If I didn't mention anything about the half-life, then you just go with the calculation, considering that you need to cover 5 half-lives of the drug. Okay? When we're talking about area under the curve, again, we're using concentration in the plasma in both the intravascular and extravascular. When we are talking about urine data, we are using the cumulative amount excreted in the urine, again, because we are using urine data versus time. So it's like I'm comparing this profile to this profile. Again, we can't reach this point, which is time of infinity for each one of them, but we can do that until certain time that I know that it's covering around 5 half-lives. Then I can compare whatever is happening at this point for the extravascular over this point. Okay? For the absolute bioavailability, my reference standard is always your IV, and it's called F sometimes, and just F without F absolute, okay? And it's the unit of the absolute bioavailability as its fraction or ratio of area under the curve, or area over area under the curve, of, or amount excreted in the urine over amount excreted in the urine. So it's the unit of it will be unitless. We may have it as percentage, so just multiply it with 100%. So the question is, can the absolute bioavailability be equal to 1? Yes, it can, because the area under the curve or amount excreted in the urine could be equal or similar to whatever we have in the IV. could be equal to less than 1. Yes, it could be, which is the usual case. It's less than the IV bolus. It could be more than 1. No, because no extravascular will have more extended rate of drug reaching the systemic circulation more than the IV bolus. Okay. For the comparative or relative bioavailability, and this we call it as F relative or F comparative. From the name, we can determine that it's relatively to something else. It's our standard. It's not anymore as the IV. It's any other extravascular or any other route of administration. So we either use the plasma data using area under the curve or urine data using the amount excreted in the urine with whatever just I mentioned before, but your standard is different. And this is measured for two different dosage forms, two different routes of administration. Usually, when we are comparing in the relative bioavailability, the generic compared to the standard or compared to the innovator. Okay, using the plasma data, which similar to what I mentioned, using the plasma data, we are using area under the curve, but the idea is I'm not talking about in reference to IV. No, now we are using different, either as we said, different drug, different, as it's mentioned here, different dosage form or route of administration. So we are talking about this different dosage form here, or different same drug with different dosage form, different route of administration as it's here. Okay, and again, each one of them is corrected to the dose of its exact drug or route of administration or dosage form. But notice that it's multiplied here, not division. That's why we just flip it, okay, because it's multiplied too, but it's the same as before. Okay, so the only difference, it's the same as for the absolute bioavailability, but the only difference that we are not using IV as a reference or a standard. We are using different extravascular a dosage form or route of administration. It's if we are talking about profiles, CB versus time, it's the same as we are comparing this to that or this to that. It doesn't matter. It's just two different extravascular. So either we are comparing different route of administration, like oral to sub-Q or intramuscular in here, or different dosage form for the same extravascular, which means we are talking, as I just mentioned in the previous slide, we are talking about oral. Both of them are oral, but one is suspension and one is tablet, or one is tablet and one is capsules, or different formulation for the same dosage form, which means that we are talking about tablet 1 and tablet 2, but we are changing the formulation in it. So we are comparing two kind of things, and we are seeing if they are what is the bioavailability, but it's called relative because it's relative to other extravascular. Using the urinary data, it's similar thing to whatever I mentioned. Again, it's urinary data, so I need to use the amount excreted in the urine total. We may not have it total, so we use 5 half-lives, time covering 5 half-lives. Again, for different extravascular, we don't have the IV as the standard. We have either different route of administration, different dosage form, different route of administration, or different formulation. So again, when we having the amount in the urine, similar again to the cases that you have, but we are talking about cumulative mass excreted in the urine. So as I'm comparing this to that, or that, or this to that. Okay. So the reference in the standard is not the IV anymore, and could be any other extravascular drug. The relative bioavailability could be equal to 1. Yes, it depends on the whatever that we are comparing. Less than 1, yes, for sure. More than 1, yes, for sure. Because less than 1, when we are comparing one formulation to another formulation, and this formulation is not giving me good bioavailability compared to this. More than 1, like example, I'm comparing the bioavailability of IM over oral, right? Or some sub-Q over IM, doesn't matter. And this is the first one that I'm comparing to is better than the second one. So yes, we can have it equal to 1, less than 1, or more than 1. The bioequivalence study, we said that to determine the bioequivalence study, we are talking about bioequivalent product, or we need to determine if the two products are bioequivalent. It's a type of relative bioavailability studies in which we are having or comparing the parameters that we are talking about in the kinetic for different products, but they are for sure equivalent product. Similar to the diagram we mentioned, it's equivalent product, and we need to know if they are bioequivalent. So we need to have no significant difference in the amount and rate of the drug reaching systemic circulation, comparing different parameters kinetic wise, but the only difference that we are comparing it to the standard or the innovator. Let's say we have a generic product that we produce in Jordan by Al-Hikmah, let's say, the Hikmah manufacturing company, compared to a drug that is the same drug with dosage form that is produced by, let's say, Sanofi. Okay, so we are comparing it to the innovator or the standard or in the market, the first one who produced the product. So the bioavailability, bioequivalence is type of the relative bioavailability, but again for equivalent products, so should be equivalent product, but the standard should be the innovator or the standard or the reference that we have in the market. And this is what is usually done by the our companies in Jordan, in which we have centers to determine if the drug that is produced by the companies is bioequivalent to the innovator. Okay, and I'll stop this recording